infection of Paramecium caudatum by Holospora
Undulata.
And remember, all undergraduates, you can stay after this session. We want to get a picture of all the undergraduate presenters that were here.
Okay, so just to begin with some background on Holospora, Holospora are bacterial endosymbionts, and there are 10 species, 6 of which can actually infect Paramecium. And from those species that can infect Paramecium, they can infect the micronucleus, the macronucleus, or both the mic and the mac of Paramecium. But for my experiments, I've been using Holospora undulata, and those are specific to the micronucleus of only Paramecium caudatum. And Paramecium are freshwater ciliates that I hope you've all learned enough about over the past few days in the Ciliate lectures if you've been attending. But so Holospora, their closest relative are Rickettsia, which are intracellular obligate pathogens. And as you can see from this figure, all of the microbes that are highlighted in red are actually microbes that exploit the nucleus of their host. And both Rickettsia and Holospora fall under that category. And both Rickettsia and Holospora, as you can see, are considered to be alpha proteobacteria and are closely related. But we hope to use the relationship between Rickettsia and Holospora to our advantage because we know Rickettsia actually has a very similar infection process to Holospora. So we hope to use the past experiments that have been done in gene expression experiments from infection of Rickettsia and their hosts and compare the genes that have been upregulated and downregulated to the genes that are upregulated and downregulated in infection experiments from Holospora and Paramecium. So to give you an overview of the Holospora life cycle, it begins with a lysate being added, and the lysate's just made out of a strain of Paramecium that's already infected because Holospora are obligate pathogens. And after 10 minutes after the addition of the lysate, you can see here that the Holospora is being taken into the oral apparatus of Holospora or of Paramecium by phagocytosis. And after approximately 30 minutes, you can see the Holospora here moving through the cytoplasm after it's broken out of the acidified food vacuole towards the micronucleus. And after 24 hours, right here, the Holospora has actually already it's already penetrated the micronucleus, but the way it does so is it uses what's called an invasion tip, and the invasion tip is made out of proteins that allows it to penetrate the micronucleus. And once it's already in the micronucleus, the infectious form is able to undergo binary fission. And you can see that the infectious form actually transforms into reproductive forms. And the reproductive forms actually begin to fill the micronucleus. And as you can see here in this picture, the micronucleus is definitely enlarged compared to normal. The micronucleus is definitely very small compared to the macronucleus normally. But this picture was taken after starvation was induced, so 7 days after the Paramecium cells were fed. And after starvation is induced and infected Paramecium, the reproductive forms, which you can see on the sides here, actually begin to transform back into infectious forms. So these are actually infectious forms over here. And after all of the reproductive forms turn back into infectious forms, they're able to break out of the micronucleus and restart the life cycle. So there have been a lot of experiments already testing how the presence of Holospora affects the host, but most notably, they found that the presence of Holospora actually induces heat shock resistance and osmotic stress resistance in the host. And They've also found that the presence of Holospora reduces or completely stops reproduction of the host, and this is especially true in species of Holospora that infect the micronucleus. And this is because the micronucleus is actually the germline nucleus in Paramecium, and it's kind of been annoying in our project because we're actually using a strain of Holospora that infect the micronucleus. They've also found that the presence of Holospora actually increases the presence of other bacteria and endocytobionts within Paramecium as well. So you can see here, this is actually, might be a little hard to see, but on the sides, there's a lot of food vacuoles in Paramecium, and Paramecium uptake a lot of bacterial in the first place. but it makes it a little hard to differentiate what is Holospora and actually what's other bacteria. But on the sides are food vacuoles that are filled with bacteria, and it's easy to differentiate it once you find the macronucleus, which is pretty large, and then trying to find the micronucleus, which is next to the macronucleus, and determining where the reproductive or infectious forms are within the mic. So as for evidence of host gene alterations in the past, there have been gene expression experiments done on rickettsia infection in hosts, but most of the gene expression experiments focused on gene expression in the bacteria itself, so in the rickettsia. But what they actually have found for gene expression in the host was a lot of actin-based activity, especially genes encoding for proteins like VAST, vinculin, and profilin. And these genes actually help with actin elongation, cell adhesion, and binding. But for gene expression experiments from Holospora obtusa infecting Paramecium, they found the specific heat shock expression protein or genes, HSP60 and HSP70, but we don't expect to find this within our experiments because we're actually focusing more on the earlier time points rather than the later time points where these heat shock expression genes are normally upregulated. So what I've actually been trying to do is trying to find what genes are up and down regulated at early time points of infection of only the susceptible strains of Paramecium caudatum by infection of Holospora undulata. And eventually, I would like to also run gene expression experiments on resistant strains of Paramecium caudatum and compare them to the gene expressions of the susceptible strains. And this is because I want to determine what genes actually cause resistance of Holospora infection in Paramecium. So what I've actually done to do all of this is I've extracted RNA from infected Paramecium species at 0 minutes, 10 minutes, and 30 minutes, the early time points after initial infection. And initial infection is just when the lysate is added at 0 minutes. And but other than that, we actually ran a mock-infected control, and the mock-infected control is just when Paramecium cells are treated with a lysate without any Holospora or an uninfected Paramecium. So the reason we did this was for two reasons. The first being we wanted to see if there are any genes that were upregulated because of just the presence of the lysate rather than the presence of Holospora themselves. But we also wanted to test for what we call the feeding effect, and this is because during my experiments, I also feed the Paramecium at 0 minutes. So I wanted to see if any Paramecium are reacting in response to the addition of the media as well. And all of these experiments were followed by cDNA library preparation and Illumina sequencing. So we're still in preliminary analysis right now, but what Jeff Gu has found from our gene expression experiments this is a graph showing the expression level changes at 0 and 30 minutes, and the X and Y axes show the log2 FPKM values. But as you can see from the diagonal, there are not many genes that stray from it, and this shows that there are not many changes in the transcriptome from 0 and 30 minutes. And here you can see that there are not many significantly upregulated genes at 0 and 30 minutes. There's actually only 45, with more being downregulated than there are significant genes being upregulated. But this analysis actually does not control for the it doesn't account for the mock-infected control. So after we wanted to try to do a better analysis to try to account for the mock-infected control. And the way we did this was we actually subtracted the log fold change from the mock-infected control from the log fold change of the initial infection. So what we got from this analysis is we determined that there's 106 upregulated genes at 30 minutes and 28 downregulated genes. But from those genes, only 20% of them actually had a functional annotation, which we think is a little strange because we know 9,000 out of 18,000 Paramecium caudatum genes actually have a functional annotation. So we believe that the 80% that don't have a functional annotation are actually Paramecium specific. We also did an analysis at 10 minutes, and we found a couple of genes that are of interest to us, especially the mate efflux family protein. And this is a multi-antimicrobial extrusion protein, and it actually extrudes cytotoxic compounds from the cell surface. And this uses an ATP or a proton-dependent process to do so. But we also are very interested in the homolog of Paramecium tetraralia 51A surface antigen protein. And we know this is a mutually exclusive protein, meaning that only one can be present on the cell surface at a time. But the reason we're actually interested in it is because it's controlled at a transcriptional level, but by environmental stresses. So we think that this is being upregulated because of the presence of Holospora. So what we can conclude from this is, especially from the 0 and the 30-minute time points, is that Paramecium are evolving in response to the presence of Holospora, especially the susceptible ones. But we think this because that we found 80% of the genes in the 30-minute time point that did not have a functional annotation, which are probably Paramecium-specific genes that are evolving at a quick pace. So what we hope to do in the future is repeat these experiments with resistant strains of Paramecium caudatum so we can compare it to the susceptible strains, and then extract RNA from not only the host but the bacteria Holospora during the infections of susceptible and resistant strains. But we also need to control for our feeding effect by simply just feeding the cells that are going to be infected before the experiment. And I would like to acknowledge Lydia Bright, Tom Doak, and Jeff Gu, specifically from my lab, who have helped me a lot in my project, and NSF for funding us, and Oliver Kaltz from the University of Montpellier for providing our Paramecium. Thank you. Any questions?
That was a nice talk.
Thanks.
I just, after looking at your fellow lab mates and your data, I'm wondering whether RNA-seq of steady-state levels of RNA is the most effective way to look at these changes, because these are large cells with potentially large stores of RNA, and whether looking at more changes in transcription rates, which there's ways to do as opposed to looking at steady-state levels, you may see more extensive changes.
Yeah, we're definitely considering changes to our analysis. This is definitely the beginning steps of our analysis. So, but especially because we're trying to control for the mock-infected control. So, yes, we'll definitely consider different analyses in the future.
I apologize if I missed it.
Yeah.
Holospora, when it's infecting, I heard you use the word symbiotic. Does it persist without? It doesn't kill the paramecium. It keeps the paramecium can live with these things inside them, right?
Yes, but in some it can decrease their fitness.
And the second question is the bias here. Is it has anyone shown this to infect other ciliates like Tetrahymena?
I don't. Yeah, that's definitely.
So TET would have its own if there was one.
Yeah.
Okay. Thank you. It was good.
Are there any plans to keep the gene expression studies going longer into infections? So, I mean, especially when they're just blasting out of the mic, and there's got to be some interesting stuff happening there.
Oh yeah, definitely. So the reason I didn't include it is because our biological replicates were a little iffy in our current analyses, but I have done RNA extractions at 60 minutes, 24 hours, 48 hours, in a week, because I'm really interested in the later time points as well.